We are definitely on the downhill slide now. So this is the chapter 18 cell cycle, and this is going to really help us to understand what's going on when we get to cancer. So a lot of this is going to be review. You guys have been learning the phases of the cell cycle for quite a while now. Probably started in 7th grade the first time you memorized them. So we'll do a little bit of review and then get into some more molecular definitions of what's going on in the control of cell cycle. So why do we have cell division? The reasons for cell division are pretty obvious. There needs to be a cell cycle duplication of cells for growth and development, for tissue renewal, for wound healing, and then single-celled organisms. This is how they reproduce. So cell cycle is every single eukaryotic cell undergoes the same phases of cell cycle. The mechanics of it may be a little bit different for different organisms, but they all go through the same phases. So we're going to talk about what are the phases of cell cycle and what happens when they occur. We're going to talk about what happens during interphase, the gap 1, the synthesis phase, gap 2, and then we're going to talk about what happens during M phase, the phases of mitosis as well as cytokinesis. We're going to talk about how those are regulated, and we're going to pay special attention to the role of cytoskeleton during the phases of mitosis. So what are the phases of the cell cycle, and in what order do they occur? What happens during the different phases? The mnemonic that I usually teach here is PPMAT. So if you just remember PPMAT, that's prophase, prometaphase, metaphase, anaphase, and telophase. Those are the mitotic phases. They all happen during M phase. M phase includes mitosis, which is the division of the chromosomes, separating the chromosomes into the two different sides of the cell. This is important because aneuploid cells, at least in animals, that's a big no-go. And then cytokinesis is also part of M phase, the division of everything else besides the chromosomes, the cellular contents. So before we can get to M phase, we have to go through the interphase, G1, a growth phase where after a cell division, we've got two fairly small cells. So they have to grow a little bit before they're able to divide, and the conditions around them has to be right. And so there's a G1 to S checkpoint that says, all right, now go into cell division. Once you pass the G1 to S checkpoint, you are committed to complete the cell cycle or else commit suicide, because during S phase, the chromosomes are duplicated, and so you would be tetraploid, and again, tetraploid is a no-go. You can't survive as a tetraploid cell, so you have to complete the cell cycle. During S phase, the centrioles are also duplicated, and that's going to be important for setting up the spindle pole for the division of the chromosomes to either side of the cell. So during S phase, we double our DNA content, and we double the number of centrioles that we have. So then after after S phase, then we'll go into a second gap phase. This is also a growth phase, but here we're doing things that are more specific to cell cycle, so producing all the proteins that are going to be required for M phase. So so during G1, this is to take the small cell that resulted from the last mitosis and grow it big enough that it's able to do its job. So so G1 is also where most cells hang out because they're doing their job. We often call that the G0, where they exit the cell cycle and they just do whatever tissue-specific things that they do. Some cells spend their entire life in G0, like neurons. And then G2 is more specific to creating the proteins that are necessary for mitosis, like microtubules, for instance. Okay, so so then after the G2 to M transition, another control point. After you do that, basically that checkpoint is it was the genetic material replicated or duplicated faithfully? Do we have all of the DNA content for cell division? Then we'll go into M phase, and M phase has, like I said, the PPMAT, PPMAT. What happens during prophase, prometaphase, metaphase, anaphase, and telophase? So what happens during prophase is nuclear envelope breakdown to provide access of the during prophase is condensation of the chromosomes, and then during prometaphase is nuclear envelope breakdown, providing access to the chromosomes, or to the kinetochores, to the microtubules, allowing the chromosomes to attach to microtubules. And then a push and pull is created by the microtubules growing at the plus end, shrinking at the plus end, in order to align the chromosomes at the metaphase plate. And so when all the chromosomes are aligned at the metaphase plate, we call that metaphase. Then once every single kinetochore is attached to a microtubule, then and only then does MAD2-BUB-R1 release from the kinetochore and activate separase. And separase is a enzyme that will cut the connections between the different sister chromatids. So we will cut cohesin and condensin and sororin, so that the dynamic tension that is created in metaphase results in chromosome movement toward the two different poles. Also during anaphase, because there are other microtubules that are not kinetochore associated, they just overlap with each other, and kinesins in between those microtubules will create a push, and so the two centrioles will move away from each other. That kind of elongates the cell. And then there are also dyneins on the aster on the other side of the centrioles, pulling on the microtubules, and so there's a pull of the two poles away from each other. So the spindle elongates due to push of the kinesins on the overlapping microtubules, a pull by dyneins on the opposite side of the centrioles, and that creates an elongation of the cells. And then when separase cuts the connections between the sister chromatids, then we see the chromosomes fly apart from each other. We also, at the end of anaphase, start to see the very first puckering of the cleavage furrow, but the cleavage furrow becomes much more obvious during telophase. The cleavage furrow will happen exactly wherever the metaphase plate is. You can actually even go in and manipulate the spindle such that it's in a different place, and then the cleavage furrow will move to the middle of the new spindle pole. So where the cleavage furrow is defined by the placement of the spindle. The cleavage furrow tightens or gets more defined as telophase progresses, and then also during telophase the nuclear envelope will reform around the new chromosomes. And then at the end of metaphase, cytokinesis happens to give you two new daughter cells from the original cell. So just to go through that one more time, prophase, we have condensation of the chromosomes, and the two centrioles will start moving to opposite ends of the cells to create or start to form the mitotic spindle. During prometaphase, we have nuclear envelope breakdown, which provides access to the kinetochores of those microtubules. So nuclear envelope breakdown and the establishment of the spindle. Then push and pull dynamic tension is created from growing and shrinking of the microtubules to align all of the chromosomes at the metaphase plate. So once they're all aligned at the metaphase plate, that will define the metaphase. You see them all lined up with their ends pointed outwards from the metaphase plate. Once every single kinetochore has a microtubule attached to it, separase becomes active, cuts the connexins between the sister chromatids, and we see the chromosomes move away from the metaphase plate to the opposite ends of the spindle pole. So now the ends of the chromosomes are pointed more toward the center of the cell, toward the metaphase plate. And we also see an elongation of the cell. The spindle poles will move away from each other due to the kinesins on the overlapping microtubules and the pull of the dyneins on the aster. Then in telophase, we start to see the contractile ring forming. So the cleavage furrow becomes obvious, and we also start to see reformation of the nuclear envelope around the chromosomes, and then finally during cytokinesis, we see the two cells get separated from each other by the contractile ring completing the cleavage furrow, and cytokinesis results in the separation of the two cells into two daughter cells, and end phase is complete. In plants, we still see the same things: prometaphase, prophase, prometaphase, metaphase, anaphase, and telophase. So we see that in plants as well as in animals. Okay, move on to the next video.